Hello, welcome to this video. Our topic today is enzymes. You would have already heard about enzymes. In your biology classes also, you would have learned about them. When you learned about biological life processes like digestion and respiration in animals and photosynthesis and respiration in plants, you would definitely have heard about enzymes. Enzymes play a very important role in digestion of nutrients in our own body. Life processes cannot proceed without enzyme catalyzed reactions. Let us see what these enzymes are. These enzymes are proteins. They are macromolecules. What do you mean by macromolecules? They have very large molecular mass. Their size is very big, so that their surface area is very great and reactants can adhere onto them when they act as catalysts in biological medium. That is why they are called biological catalysts. They are called biological catalysts. Physically, they are colloids. We will be learning about colloids in the next video. Now, let us see a few examples of enzyme action. I have already prepared a chart here. You may also prepare one like this. Invertase, present in yeast, can convert sucrose to glucose and fructose. Cymase, also present in yeast. It converts glucose to ethyl alcohol and carbon dioxide. This reaction is familiar to you. This is fermentation. Diastase, present in malt, converts starch to maltose. Maltase, also present in yeast, converts maltose to glucose. Urease, another enzyme present in soybean, can convert urea to ammonia and carbon dioxide. Pepsin, it is an enzyme present in gastric juice secreted by the stomach, and it can convert protein to amino acids. Lactic acid bacteria secretes lactobacilli enzyme. It is present in curd. It can convert milk to curd. In our houses, we convert milk to curd by adding a little amount of curd to that milk, because this curd contains this enzyme due to the presence of the bacteria. It is a useful bacteria. Now, pancreatic trypsin. It is an enzyme present in pancreatic juice, and the pancreatic juice is secreted into the intestine. It can convert protein to amino acids. I have given here some 8 examples. You can try to remember these examples. The substance on which the enzyme acts is called substrate. In this case, sucrose is the substrate. In the second case, glucose is the substrate, and so on. Now, while discussing this, I have not given the chemical formula of the substrate or the products that are formed. That is only to make the portion a little bit easy. Okay. Now, let us see the features of enzymes. Just like catalysts, enzymes also have certain features. Many of the features resemble that of catalysts because they are, after all, these enzymes are, after all, biological catalysts. They also have high specificity. Any example can be given here. For example, diastase can convert only starch to maltose. Maltase can convert maltose to glucose. So, there is much specificity in their action. They are needed only in small quantities because they are regenerated. They need optimum pH. What is pH? The pH value of a medium tells us about its acidity or basicity, that is how far it is acidic or how far it is basic. The pH scale consists of reading 0 to 14 and 7, the reading 7 is neutral point. The optimum pH which is favorable for the action of enzyme is somewhere between 5 and 7. 7.1 or 7.2. That means slightly acidic or slightly alkaline nature. Sometimes neutral medium also seem to facilitate the action of enzyme. Optimum temperature is another feature. It requires optimum temperature. What do you mean by optimum? And the temperature should not be too low, it should not be too high. It should never be too high because enzymes are after all proteins and these proteins get denatured. They lose their property when the temperature becomes very high. Activators and coenzymes promote the activity of the enzyme. The activators are metallic ions attached to the enzyme and coenzymes are non-protein part of the enzyme. In most cases they are certain vitamins. Certain vitamins are very essential for the proper functioning of enzymes. Inhibitors reduce the activity of the enzyme. Say for example, drugs. Drugs are very dangerous things. They act on the enzyme which maintain life processes and inhibits its function, prevents it from acting. So they are very harmful for the body. Now let us see a comparison of catalytic reaction and enzymatic reaction. The functioning is more or less the same, but there is some difference in the terms that we use here. Say for example, in this case, in the case of catalytic reaction, we use the word promoter. Whereas in the case of enzyme, we use the word activator. For poisons, in catalytic action, we use the word inhibitor. In enzymatic action, catalysts have free valency. Whereas here we talk about active sites. Or activation centers. Here, catalytic reaction, we use the word intermediate complex. Intermediate complex. Here we use the term activation complex or activated complex. Now, the reactants in the case of catalytic action are called adsorbates. Here we use the word substrate. Now, let us see mechanism of enzyme action. The enzyme fits into the substrate or vice versa. Substrate fits into the enzyme. How is this made possible? The enzymes have certain depressions on its surface. In this diagrammatic representation, I have shown only one or two depressions and one projection. There may be a large number of such depressions and projections. So, the enzyme has a particular shape due to its projections and depressions. The substrate also has complementary shape. so that these depressions and projections can fit into the substrate. So the enzyme is something like a key, and the substrate we can say is something like a lock. Just as one key is specific for one particular lock, one particular enzyme is specific for one particular substrate. When the enzyme and substrate attach like this, let us represent that in the form of an equation. Enzyme plus substrate gives enzyme substrate complex. This is the activation complex. In the next step, enzyme substrate complex splits into enzyme and product. P stands for product. This is the enzyme. It is freed, and this is the product. So this is the mechanism of enzyme action. Questions may come from mechanism of enzyme action. You may also be asked to compare catalytic action and enzymatic action. It is more or less the same, but what you should remember here is there are certain differences in the terms that are used. Okay, a small portion which remains to be covered here is industrial catalytic process. I have already discussed that portion in the previous videos. You can just refer video 7 and 8 where I have mentioned this catalyst functioning in industrial process. Say Haber's process is an industrial process for synthesis of ammonia. Contact process is an industrial process for synthesis of sulfuric acid. Then there is Oswald process, which is again an industrial process for synthesis of nitric acid. Such processes I have already discussed there. So I am not discussing the details again in this video. You can go back to that video and see. I hope this video was useful to you. In the next video, we will take up colloids. Thank you.